Hi, everyone. So excited that you could join us for our third Future of Food series at Bluestein. This time, we are talking about all things microbiome. I've got two amazing panelists with me here today. We've got Kevin Honaker and Jack Gilbert, whom I will introduce to you soon. The plan for today is we'll talk about BiomeSense, a little bit more about Bluestein Ventures, and then we'll really dig into all things microbiome. If you could, throughout our conversation, please drop your questions into the chat function. We will be leaving time at the end for some Q&A with Jack and Kevin as well. I will take it off by introducing myself, and then love to hand it over to Kevin and Jack to discuss a bit more about their backgrounds and their role at BiomeSense. So I'll introduce myself first. I am Amira Khatib. I am a senior associate at Bluestein Ventures. Bluestein Ventures is an early stage VC firm based in Chicago that invests all across the food value chain. We like to say that we invest in goods on the shelf, how goods get to the shelf, and how goods are made. Bluestein is also an investor in BiomeSense and has been since 2020. And for those who aren't familiar, and we'll hear a lot about BiomeSense today from Jack and Kevin, BiomeSense is disrupting the $100 billion microbiome precision medicine market by developing the first technology platform to enable continuous standardized measurement and analysis of microbiome. So today I have the pleasure of introducing Jack Gilbert. Jack is one of the preeminent thought leaders in the microbiome space and a co-founder of the Earth Microbiome Project and American Gut Project. He also serves as a scientific advisor to BiomeSense. Also joined by Kevin Honaker, who is the CEO and co-founder of BiomeSense. And I will let Kevin kick it off and share a bit more about himself and where BiomeSense is at today. And then I'll let Jack jump in as well.
Yeah, great. Thank you. So I'll just quickly introduce myself. So yeah, Kevin Honaker, co-founder and CEO of BiomeSense. So quick background on me. I have just over 10 years now, life science and healthcare experience, working in pretty much all aspects of the healthcare value chain, everything from provider, life sciences, a few years in insurance, believe it or not, so touch all that bit. And then have spent most of my time over the past however many years on early stage life sciences, commercialization, therapeutics, diagnostics, and precision medicine. And that's my true passion, and what I'm really excited at BiomeSense is accelerating precision medicine in general, and the microbiome in particular's role for that. I came from pharmacogenomics before this, genomic precision medicine, and now, really hope that we can help bring and make the microbiome that true pillar of precision medicine that we know it can be. So quickly, where BiomeSense is today. So it's been a long journey. We've founded this company, I think, 4 or 5 years ago now, alongside Dr. Gilbert, Jack here. And so we started, it was basic, in many ways, me, Jack, an idea. And then ever since then, we've raised a bunch of money, got the team in place, have our technology up. We'll talk a little bit more about it later. It's deployed in homes, right, generating data right now. And we're essentially raising a little bit of money right now to then take it through to commercial launch. We got plenty of partners lined up, and I'm going to go into a lot more detail later, so I'll skip that. But yeah, so that's where we are today.
All right. Thanks, Kevin. My name's Jack Gilbert. I'm a professor of pediatrics at University of California, San Diego, down here on the Southwest Coast. I have been in academic and industrial science for about 20 years. I've worked for Unilever, University of Queen's University in Canada, Plymouth Marine Laboratory in the United Kingdom, and then also University of Chicago and Argonne National Labs, where I met Kevin. From 2010 to about 2019. So I moved here in 2019 and currently am principal investigator of the Microbiome and Metagenomics Center for the $175 million NIH Nutrition for Precision Health program, where we deploy innovative microbiome-based technologies to understand how bacteria inside your stool, inside your gut, are associated or can be predictive of how your body will respond to diet. So this is key for understanding a number of different diseases, everything from diabetes to cardiovascular and cardiometabolic diseases, even to neurological diseases such as gut-brain health, in depression, anxiety, and neurodegenerative disorders. And so we have a wide portfolio of research here where the gut lab, this BiomeSense technology, is being deployed.
Amazing. Thank you both. Jack, would love to start with you and just kind of start with some of the basics to make sure that we're all on the same page. So microbiome and gut health are very trendy words, but you know, the microbiome is kind of a nebulous concept. So can you help us understand first, what is the microbiome and what role does it play in our overall health?
Yeah, it's interesting. In science, it's trendy as well, which is funny, right? You know, the zeitgeist has led us to use this phrase, but the phrase really is another word for what we term microbial ecology. Now, microbes are bacteria, archaea, tiny little unicellular things that run around, viruses, small fungi, even protozoa, which are tiny little plants and animals, right, that run around inside the oceans, inside your body, everywhere. So these microscopic organisms live in many different environments, and in human health, they tend to inhabit our intestines, our skin, our urogenital tract, your bladder, our lungs, our mouths, our eyes. So they're all over us, basically. You have around 40 trillion bacteria living in or on you. Most of it's in your intestine, in your colon, in your duodenum, your small bowel. And that mass, maybe about 0.5 kilogram to 1 kilogram of microbial biomass, is responsible for helping to process your food. So they actually ferment fiber, which your body can't break down. So when you eat plants, they're the things that take all the nutrients out of the plants and provide it to your body. They also release, in the process of being alive, some of their excretion products our bodies have become dependent on. So they release chemicals which get inside our body, feed our cells, help our mitochondria, the little powerhouses that run our cells, help them to function properly, help our bones and our even our muscles to become productive. When we remove those signals, we can get osteoporosis and myopenia, loss of muscle tissue. They can even regulate how our brains work, right? So altering signals between the gut and the brain, or changing our inflammation state to make our brain work in a different way. So we found over the last 15 years of research in hundreds of cohorts that using some really rudimentary techniques, we can discover some incredible low-hanging fruit in terms of how microbes influence human health. And now we're moving into this next phase where we've discovered that really to understand or to identify the key organisms, key microbes that associate with some of these health trends, we really need to look over time. We need a detailed investigation of how things fluctuate day to day, changes with different diets. And using that kind of approach, we can start to really dig down deep into the features of the microbiome, the species, their functions that are key for driving human health therapeutic benefit.
Yeah, I think that's a great kind of segue, Kevin, for a question I'd love to get your perspective on. So we know there's a lot of promise around the microbiome, but also many challenges, and there's a huge issue around reliability of testing and kind of limited quality data within the industry. Can you tell us a little bit more about how BiomeSense is solving these problems and kind of the role in the industry?
Yeah, no, and I think as Jack mentioned, the microbiome is such huge potential, right? And we've seen it with different clinical implementations of it, like, really have that impact. But yeah, the reliability of testing and the reliability of measurement, right, is an enormous problem. An analogy I like to use to kind of talk about why, right, is like, kind of imagine you went to your doctor tomorrow and they checked your blood pressure, right? They had some tool, they did it, and they said, your blood pressure's super high. You got to take a drug to lower it. That's what we need to do. But you know what? This tool's been a little janky. I'm going to bring in a second doctor. So the second doctor walks in the room. She tests it with her own tool, and she looks at her chart and says, wait a minute, no, this is really low. You need a drug to increase it, right? And then now you're confused. So a third one walks in the room with a third tool and says, no, I checked it. I get a different number. And according to my chart, you're totally fine, right? Good to go. And if you're sitting there, right, well, it can't work that way. Healthcare doesn't work that way. Really don't have anything that you can use, right? But in many ways, the microbiome suffers from issues like this because measuring it reliably is extremely difficult to do. And Jack's lab and Jack's colleague Rob have invested, I don't even know how much money, time, and effort, right, into the lab.
Hundreds of millions.
To doing it right, because that's what it takes to do it using existing techniques, right? But most people don't do that. And that's why you'll see in paper after paper has shown this, different labs give different answers, different companies give different results, right? It can't be consistent. And of course, the longitudinal aspect, like Jack mentioned, right? So BiomeSense looked at this and we said, look, if the microbiome is going to take off and we want to think about what is it when it's in the clinic, right? And for that, we have to be able to reliably, accurately, and completely measure it, right? Because that is what it's going to take. And so, but that's really, really hard to do. And so that's what we actually did is we built the very first end-to-end sample to answer technology platform to make continuous, standardized, and reliable measurement and analysis of the human gut microbiome. And so with the goal, what's so exciting about this is like, it's not just something that, for the consumer, like in the doctor's office, right? But things that scientists, right, and people in the space who are developing these drugs, products, right, and other things with the microbiome can use to understand the microbiome to a depth and level that has never before been possible. And once you have that understanding, we think about everything Jack said about how exciting it is based on what we know now. Well, imagine what we're going to be able to do once we fully unlock and understand it right to that level. And really, that is what BiomeSense is doing, is deploying this technology and creating this large standardized database at a scale far beyond anything that's existed in the microbiome field before, to give to scientists like Jack, right, to take that and essentially allow us to unlock the microbiome and really accelerate the field forward. And then long term, yeah, we also want to be something the consumer uses and the clinician uses as well, right? And obviously, we'll talk a little bit of personalized nutrition, which is a big personal thing of mine, so I'm really excited to talk about that. But yeah.
Yeah, I guess to that point, can you talk a little bit more about some of the challenges and opportunities with what you and the team are building at BiomeSense?
Yeah, so basically, like the challenges with the measurement thing, right? It's not like people get different answers because they're evil or bad or like, dumb. It's like I said, it's really hard to do. And that's the problem, right? And the reason for this is because you have technical and scientific challenges with, these are microbes as Jack described, right? So if I'm trying to get a sample, often it's stool samples, right? And trying to get those to a lab, I've got to preserve them. If I screw that up, they're going to grow and it's going to change, right? I got to process it in the lab, but these are different cell types. This is a very difficult sample to work with, very prone to error, right? And so that entire pipeline is very prone to error and very difficult to do properly. And then the other part is, well, I want to be sampling you continuously because I want to see how your microbiome is changing over time, like your heart rate or blood pressure, right? As Jack was talking about. But stool samples, for people on the call, who've seen this, right? It's not a fun process. It is very, very, very unpleasant, right? And it's something that people don't want to do. I like to give the analogy of like the, continuous glucose monitors versus like pricking the finger. Like there were diabetics would literally get amputated because they wouldn't be willing to prick their finger 4 times a day because that was so unpleasant, right? And then you think about, you switch to the continuous glucose monitors, right? And a lot of that problem went away because people could continuously track. So that patient facing part of it or consumer facing part is critical. So you've got a huge technical challenge and a user challenge, right? And so our solution to that, there's no analog as far as I know it. You can see it behind me. It's our gut lab technology. And basically we said, let's do the best of both worlds. Let's put a fully automated miniature laboratory that has been technically validated, fully automated in the home, right? Or in the clinical site or wherever it needs to go to enable continuous and standardized tracking of the microbiome. So solving both problems at once, right? So not only we get the same answer every single time, but we're also able to track how that microbiome grows and changes over time, making it easy to use, right? And really allowing this to be information that could be used by anybody, right? So like scientists like Jack, but also ultimately, as I mentioned, the clinicians and even the consumers trying to understand their microbiome, right? And leverage it for health or dietary probiotics, right? And those sort of other interventions. So yeah, so that's what we're doing. And then yeah, so actually, as long as we're here, I have a question, for you, Amira, which is like, love to know what initially got, Blue Stein interested in us, right? And BiomeSense specifically, and also, right, the microbiome in general.
Yeah, that's a great segue and great question. So at Blue Stein, we're really big believers in what we call food as medicine, and the body's ability to heal itself through food. And we know that right now we've just kind of only scratched the surface with the potential there. But at the same time, every body is unique, right? And we've been talking about that today. And that's why personalized nutrition is so critical to our individual health. And one thing that we've observed in the last few years, and I'm sure we'll talk about it more today, is this rise in what we call measured body, or the consumer's interest and willingness to track their health performance, whether that's through a CGM like levels or Oura Ring or Whoop. And as we've talked about today, we've kind of seen a number of startups in the microbiome space that either promise to reliably measure or kind of fix our guts, whether that's through supplements or products. But as investors, we've really struggled with the lack of science and data to support some of these claims, knowing that it's just hard in general to even know what's happening in our guts, given the lack of standardization. So, it's really stood out to us about BiomeSense and why we're so excited about its potential is just the potential for the business to completely transform the industry, starting with measurement and data. And, that by doing so just allows us to transform our understanding of our guts and of the microbiome, which we believe ultimately kind of holds the key to personalized nutrition as well as food as medicine for consumers. So that is why we're really excited. And I think that's a great segue as well for another question for you, Kevin. Would love to hear a little bit more about the future of the business. So BiomeSense has secured really impressive potential partners on its pre-launch waitlist, including MD Anderson, Mayo Clinic, and in some of Blue Stein's conversations with those partners, we've heard some really fascinating potential research and use cases, and would love to kind of share a little bit, hear your perspective about the types of research and breakthrough developments that you hope BiomeSense will enable in the microbiome industry.
Yeah, no, absolutely. And I think this is, for me personally, like what allows me to, as any startup when you have ups and downs, right, but to keep going is like, it's so incredibly exciting because fundamentally, right, BiomeSense is a bedrock measurement tool, right, is like, you got to measure the microbiome accurately, whether you're doing science, whether you're in the clinic or you're using it at home, right? And so we're applicable basically everywhere the microbiome is, right? And so, because that's essentially what we do. So we're looking at supporting these studies that cover almost every conceivable topic, right? Everything from neurological diseases, the gut brain, right, to GI and so on and so forth. But one of the ones I really want to talk about, because I'm most excited about, and it's relevant to this conversation, right, actually has the potential to transform food as medicine. And, to give a quick background, right, there's a certain class of drugs called immunotherapies that probably started getting really popular maybe like 10 years ago or so, give or take, right? And the reason these are such an exciting innovation, right, is because they are incredibly effective at fighting cancer. Not a lot of people know about them, right? But they basically can cure metastatic cancer. Like that's been shown, like cancers that would have been a death sentence, right? For certain populations, these drugs can literally just cure it completely, right? And it does that by essentially unlocking your immune system. However, these drugs have a huge problem with them, is that they basically work 30% of the time. And they work, and another way to put it is 30% of the time, they work really well. And then the rest of the time, they don't really work at all, right? And so right now, historically, we believe that has been due to genetics, and like a genetic test, like, is what they use to prescribe that drug. But recent research has shown that the microbiome actually potentially plays a very significant role. And they actually showed that by using fecal microbiota transplant, they could get people to respond to these drugs who would otherwise fail, which is absolutely incredible because you can't change your genes, right? So if you get a genetic test and you're done for, right? Versus this can be fixed. So this partner has effectively done some research and shown that they believe that using controlled feeding, right, and using a very specialized and strict diet for these participants, they can actually help them sort of respond to these immunotherapies, even the ones who wouldn't otherwise, by shaping the microbiome. So the combination of the food shaping the microbiome, which then shapes these outcomes, right? And this is unbelievably exciting, right? Because this is something that, we say food is medicine. We often talk from like a very loosey-goosey sort of way, right? But in this case, this would literally be a therapeutic protocol, right? Like this could be absolutely groundbreaking, game-changing for food as medicine, because this would be the sort of thing everyone who reports to a hospital with cancer, like this is what you get prescribed. It's a strict diet to change your microbiome. And all of a sudden that becomes part of the clinical care regimen, right? And now this is part of what you do. And that's sort of like, that's just one example of the sort of research that we're going to be supporting. And our role there is to track these people over time, see how it's changing, get that accurate data, see as they take the drugs, as they change their diets, what's happening in the microbiome, link that to the clinical outcomes, and be able to sort of untangle, right, what's working, what's not, what's shaping the microbiome, and allow us to, because it could be that it's not one diet, you have to change it for multiple people. But in order to figure that out, you have to have data and reliable data. And that's what BiomeSense is enabling, and super excited about what that potentially means. You could potentially use the diet and the microbiome to cure cancer is sort of the punchline there. And so, yeah, so excited.
Yeah. It's really amazing when you think about it. Would love to tie it back to the consumer as well. So Kevin, what are some of the potential applications that you see both in the near term and in the future on the consumer side? 10 years from now, are we all going to have smart toilets installed into our homes, or what do you think?
Yeah, no, absolutely. Well, I'm pretty sure Jack will. So, and hopefully I will as well. So yeah, no, I think we're definitely going that way, right? So we're obviously over the past few years, we're moving towards the quantified self, right? And like that's been very, very clear with wearables, right, and everything else. What sort of sets BiomeSense apart is that we are sort of the next gen of quantified self, because the data that we're gathering is much more advanced and much more complicated than like your heart rate, for example, right? Which is amazing, but there's only so much you can do there, right? And so right now, the way to think about it is, those microbes, they're inside you and they matter. They are influencing your health. But right now, they're just passengers. You don't really know why. You can't really do anything about it, right? And so, it's kind of like hope you get lucky, right? But that it won't be that way. And so what we're going to have is like, our goal is to put this in a smart toilet that essentially is, you don't have to do anything. You go about your day-to-day routine, right? And we're able to sort of like, as you're going to the bathroom, right? Or, potentially like oral and other sort of microbiome as well, gather all that data, be processing it in real time, right? Ping it to the cloud, right? To our analytics program. And then, so sort of like go to the bathroom morning, get up, go, get a ping to your phone, right? Hey, this is like your gut health information. This is your gut index. Like, this is what you should be looking out for, like things like that. And then you can essentially start leveraging that data for better health. And so the future is taking all these other quantifications and starting to layer on this deeper and more advanced data and moving these microbes from passengers, right, to actual integrated parts of your health that you understand, track, and ultimately leverage, right, for your diet or whatever else it is. Once we make it easy, it is going to be rapidly adopted, right? And I think that's really, the goal that we're shooting for. So.
Yeah, that's amazing. Jack, would love to turn it over to you. So, I think more broadly in the industry, unfortunately, we've seen some pretty questionable science and even some outright fraud within the microbiome space. So in your mind, kind of where does the industry stand currently? And do you think that a certain level of skepticism is warranted?
Oh, I am the biggest skeptic. I have to be. That's my job. My job is to be a skeptic. It was those first 15 years or so. The first 10 years of that were really interesting. We developed a bunch of technologies, which we obviously published in the literature and were deployed widely across the world. And companies use those technologies and abuse them to create, as you pointed out, very questionable results, and trying to provide information and advice back to consumers, which was horrendously inaccurate. And I fought against that rigorously to the point where we managed to get many of those companies shut down. So to my mind, this is an absolute this is a very important thing. You know, as scientists, we should be gatekeepers in order to make sure that the correct information, the correct details, the correct approaches and technologies are getting out there, and that they are being appropriately benchmarked, that people aren't being sold snake oil. So, you know, where are we now? Well, there are a number of enterprises, some very exciting products have got into the consumer market and into the clinical market. We have just got to the point where we have 2 clinical therapies that are about to be FDA approved. That's the first time in microbiome history that's happened. That's going to open the floodgates because now it's gone through the regulatory process, through all of the hoops and hurdles. We now are in a situation where all of the other things which have been waiting for that to happen can now start to progress through the FDA approval margin. So that will open the floodgates for microbiome therapies to become more widely accepted in clinical settings, which will improve consumer confidence, because it will improve my confidence. I'm not just a scientist, I'm also a consumer, right? So for me, that's really important to understand. You know, we're at a stage now where with a small amount of data, and I was answering these questions in the Q&A, I apologize about that, I'll answer them online. But in the consumer sector, we've seen companies come up, one of which I'm on the scientific advisory board for, who are using single time point samples of the microbiome plus some biometrics from individuals to predict their glycemic response to food, to give them a unique glycemic index on that food item. We've demonstrated, and they've demonstrated, that with a single sample, they can get about a 65–70% accuracy rating on that prediction. But with 9, 10, 12 consecutive samples, so looking at that dynamic range, they can improve that up to 85–90%. And with standardized data, it goes higher, right? Up into the 90+95% accuracy rate. That's what we want. We want to improve the accuracy of our AI-based predictions so that for immunotherapy drugs, as Kevin was talking about, for precision nutrition, for understanding and predicting your likelihood of developing a disease, such as, you know, predicting asthma. We have biomarkers now that we think can predict asthma at the age of 7 from a sample at the age of 2, right? So these kinds of trajectories we think can become incredibly important, but only with appropriate standardization. And the technology that Kevin's team has managed to develop from all of our testing seems to be incredibly stable, right? So normally, as you say, we poop in a hat, we collect some of that poop, we put it in a jar with some preservative, we seal it, we put it in a bag, or sometimes not any preservative, which is worse, and then we ship it, or we carry it in our car to a doctor's or to a company, right? And then a group of people in a laboratory use some automation, but mostly manual labor in order to try and extract the materials from that stool and use it for molecular approaches. There are so many steps along that pipeline, which we've struggled with. In all of our attempts to create statistical approaches to account for them are just masking the problem. So this new technology, this gut lab technology, removes all of that noise. You know, we don't have somebody having to scoop an exact amount of stool out of a hat. We don't have any preservative damaging the material. We don't have any shipping issues. We don't have anybody in the lab making a mistake because they didn't have their coffee that morning, right? We have a robot which does it reliably every single time without any difference. That's game changing for us, because it might put some people in my lab out of a job, but it will utterly change the paradigm for the quality of data that we can have. And with some new technologies that we're hoping BiomeSense can adopt, we'll create quantitative microbiome data. And quantitative microbiome data puts us in the spot where we can use proper artificial intelligence approaches. Not the ones that we're using now are half. There are AI light versions because they're dealing with proportional data, right? Our data is percentages of organisms, which is highly can fluctuate based upon many different variables, but absolute abundances of organisms, absolute abundances of metabolic function, that is a game changer. Now we can put it into AI approaches, which with working with companies like IBM can really push the frontiers for how this information can be made productive and predictive for healthcare scenarios.
Would love to, thanks Jack, would love to just switch gears a little bit and dive in further on the role of individualized microbiome testing in kind of developing personalized nutrition recommendations moving forward. So question for you, Kevin, where is personalized nutrition headed and what does the future look like to you?
Yeah, so I think it's headed towards a really exciting future. It's headed in the right way, right? Is like nutrition, of course, has always been personalized, right? Because not only is it how do you react to food, but it's also what is your own health goal? Like your goal with what the food you eat might be different than mine, right? And so every aspect of this is personalized, right? And it's always what are my goals and how is the food that I eat and my diet impact my progress towards those goals, right? And I don't think the first one is changing at all, but the second one is, and this is just as we get more data, more numbers, and we understand this better, right? We understand what that food is doing, right? Like, however many years ago, people didn't really even understand calorie, like your fat, grams of fat, that wasn't even a thing. Then we get calories, we get grams, we have these numbers that we quantify, right? So we get some information. And now we're discovering more and more that that's too simple. Now, glycemic response and all these other things, right? The microbiome absolutely matters. And Jack's talked about this, right? It's like, and it's always mattered. It's just, we haven't had the tools to understand it, right? Or track it or leverage it. But now we're adding that in there, right? And now we know that when the food goes in the mouth, it's not the same for everyone and that the microbiome plays this role. So we're getting to that complete picture, right? Of not just calories, not just fat, right? Or carbs or whatever you're interested in, but how that food actually, from a systemic perspective in your entire body, sort of influences you and your goals. And I think where we're going for is, the ability to not only track that information like technology like ours to build that in and keep you updated on your goals, but I think we're also moving towards, as we get more information, we're going to hit a peak where there's too much, right? And I can't follow it anymore, right? It's like way too hard to sort of do all of this. And I think what you're going to see, and I hope BiomeSense, like, is sort of one of my goals, right, is helping move, taking this complexity, getting that huge mess of data, this huge complicated data set. But then on our end, our goal as the scientists working with Jack's team, right, working with these top scientists is how do we take that information and boil it up and boil it down, right, into something usable, right? And give the consumer, like, take away that complexity and tell them what they need to know to get to where they need to be. And I think that's really sort of the future of where this is going.
Yeah. And in your mind, Kevin, what types of tools and research are really needed to get there to that future?
Yeah, I mean, it's for the microbiome specifically, right? You have to be able to measure it reliably, right? And so you absolutely have to do that, because if you can't do that, you're done, game over. Like nothing else matters, right? So technology like that, but then of course, like as I mentioned, tools and services to take that information and condense it and package it in a way that's usable and actionable and easy to access, right? Like you're not going to go look up a giant index every time, every day to see, oh, what should I eat today? Like, let me go look at this, library until I figure it out, right? It's giving you that easy to go information, right? So these technologies that will power it are going to be the ones, the infrastructure technologies that take the burden away from measuring it, right? That make it easy, low cost, scalable to measure, the burden away from analysis, right? But most importantly, and this is the part that, we don't want to skip over, is useful and actionable, right? Is I could do the first two. I could tell you that it's super easy and here's your magical microbiome number, right? And like, do this. But if it's not correct or it's not based on good science, it's not based on fun, you talk about fraud in the industry, right? Things like that, like, and that is for BiomeSense, is we are not going to go that direction. Like it's not the way we're going to go. Is we want to make sure that the information we're giving you is going to help you, make you healthy and help you hit your goals. So yeah.
Yeah, I think that's a good segue for a fun question for you, Jack. I think looking into the future, is it so far off to think that, one day we're going to be uploading our microbiome data to our Sweetgreen app so that our salad is optimized for our own personal gut health?
I mean, if it isn't, I'm heading in the wrong direction research-wise, right? That's exactly what we're pushing for. We need, in order to be able to have the biggest effect for nutrition, we need rapid turnaround, right? We can't wait. If you're having let's say we find a microbial signature in somebody who has a history or a genetic predisposition towards chronic depression, chronic major depressive disorder, right? This is a periodic disorder. It can shut you down for months. All right, what if we have identified features in the microbiome which are indicative of an onset period, right? So I can predict that problem 2 weeks out, right? That would be huge. That would be fantastic. But if it takes 2 to 3 weeks to get the results back and provide that information back on what therapy would be most appropriate, then it's pretty useless, because then you're going to be back in your depressive state again, right? You might as well stick on the randomization trials of antidepressants. So what we're hoping to do is to get that answer back to people as quickly as possible. So, working with my colleague Rob Knight, we're developing new technologies, new microprocessing chips, which can rapidly process data, not just in now hours and days, which is what it takes currently, especially for the kinds of levels of data we're getting off of Kevin's device, but down to seconds. So we can process now terabytes of data and identify, using some really fun dimensionality reduction technologies, identify microbiome traits which are associated with a particular disorder very quickly. We can only do that if we have a very, very large, very standardized database, which obviously is exactly what Kevin's talking about developing. But once we have it, this AI system should be able to take the sample, run it through a processing step, and Kevin's telling me that I can get that processing step built into his device in the next few years, and I'm hoping that's true, because there's a lot of people who would love to see that. I am one. NASA is another. The Army is another. Right? We need that technology. If we can get that all built in there so that it produces data, not just collects a sample, preserves it in an ideal condition, but produces data, then we have the technology, the microprocessing chip technology, to rapidly process that information, compare that sample against a database of millions of samples, and identify, in double-blind controlled, placebo-controlled clinical trials, all that data, and compare your sample to this gold standard database and identify that, yes, in about 2 weeks' time, you have a high likelihood of having a depressive episode. And you should start consuming more maybe out of season products, such as off the top of my head, I don't know, let's put some chai seeds in there and in your soup, and it will help. It will significantly alleviate this response, because it will cause the growth of this particular organism. You're feeding that bug, which is going to release this particular compound, maybe gamma aminobutyric acid, which will stabilize your neuropathological symptomology by altering brain chemistry, right? And everything I said there is feasible. We just need to join those dots. So yeah, give us like 5 years and well, less than that, I hope, and enough investment and anything's feasible. We just need to join those pieces together. It's an engineering problem, not a science fiction problem.
Yeah. We'll have to do a future of food series part 2 microbiome in 5 years and revisit this conversation.
Yeah, then I'll be even more ambitious, trust me. Well, I really want to answer the questions. Why am I not allowed to answer the questions?
No, you can answer. That's a great reminder for folks. We're coming close to the end of our conversation. And so if you have questions, please continue to drop those in the chat. Jack will either answer them if he has time, or we'll leave them to the end to answer live. So transitioning now to kind of a fun rapid fire Q&A section to close us out. So, as we know, consumers are really overloaded these days with how they can improve their gut health. As we talked about, gut health is super trendy and can be really overwhelming to determine what's backed by science and what's not. So with Kevin and Jack here, we thought it would be fun to do a little game of rapid fire questions on common advice to get their expert thoughts and separate what's fact from fiction for you all. So we will start with Jack. Question for you: can probiotics fix your gut, and are probiotics and prebiotic supplements and food and beverage products such as kombucha, prebiotic sodas, yogurt, a panacea, or all kind of BS?
They're not BS. We just don't necessarily understand which people it will help and which people it will potentially hinder, right? And this is the complication. For example, we just ran a clinical trial and we're analyzing the results now, looking at kombucha consumption to understand its influence upon systemic inflammation. So in this study, we randomized people to either consume kombucha, approximately 750 mils once a day, or in 2 portions a day, or to consume while they're on a standardized American diet. No offense to the Americans here, but the American diet is not our favorite diet for improving inflammation. So we're trying to see if you're in a background state where you're consuming foods which are going to make you inflamed, and you consume kombucha on top of that to stimulate microbes which could bring that inflammation down, will it work, right? And in that, we've seen variable results, right? In some people, about 75% of people, it does appear to work. In about 25% of people, it doesn't, right? What's going on there? And one of my colleagues up at Stanford did another study where he demonstrated that consumption of fermented foods in certain people appeared to promote the ability of a high fiber diet to be effective, right? So if you're going to start eating a lot of salads, a lot more plant material in your diet, for some people, they actually need to have their gut primed with a fermented food diet, be that a kimchi or a kombucha or something else that will somehow kickstart the intestinal microbial response and get those fiber fermenting organisms ready to receive that food, right? And so there's a weird piece there. Some really interesting ideas. Yes, looks like it could have some beneficial effect in certain people. And about 5% of people in our population for whom it caused not just severe bloating, but a massive inflammation episode, right? So some people, it was actually detrimental to their health in the short term, and we had to stop the trial for those people. What's going on there? This is a really good indication. Science is not perfect. We need more studies, right? More rigorous studies to really understand what's going on. And we need more studies with more long-term, longitudinal data as the Gut Lab can provide in order to really untangle this mess, right? Everybody is responding in a unique way. Precision nutrition medicine, there's a hint in the title, right? Every single person needs something slightly different. So these one-size-fits-all answers are never ideal.
Kevin, a few quick questions for you. Can exercise impact your microbiome, and can we define a healthy, normal microbiome?
Can exercise impact your microbiome? I believe some papers suggest that it can. Personally, I'm going to use the Gut Lab and find out. I'm trying to figure that out. And can you define a healthy microbiome, a healthy, normal microbiome? In aggregate, absolutely not. For you, potentially, right? And as a quick example, the healthy microbiome, if I'm trying to respond to PD-1 immunotherapies, might be very different than what a healthy one is for if I'm not trying to do that, right? So you quickly realize the idea of the healthy gut is just a myth, because it's going to depend on your goals, if nothing else. And so, yeah, so that's
awesome. Thank you so much, both. Going to turn it over. We have one question from the audience, and would love to get both of your thoughts. So, an ignorant question, it says. I'm sure it's not ignorant. No ignorant questions here.
Definitely not ignorant. This is a really important question.
Quantification is important, but can you shed light regarding what we are actually measuring / quantify from the microbiome? Like what is it that the data looks like, and give examples of how to use this quantification?
Yeah, this is the big thing, right? The problem is when we are for years, right? If we went into an environment and you wanted to count the number of, I don't know, elephants on the Serengeti, right? You would go and do randomized sampling of the Serengeti. You wouldn't count all the elephants in the Serengeti. You just count, go in and make 50 observations randomized across time and space and say, in those randomizations, I see, X percent, and I can extrapolate that percentage to the entire Serengeti and say there could be approximately 60,000 elephants, right? That's essentially what we've been doing with the microbiome for the last 15 years, is The tools and technologies that could provide quantification are too specific and too expensive to apply at a broad scale, right? Everybody's now taken an RT-qPCR test, I hope, for SARS-CoV-2. That's the kind of approach we would need, but we would need it for every single one of the 500 to 1,000 different bacteria present in your intestine every single day. So that would cost about $6 to $7 a sample. The markup on some of those tests is very large, right? But so $6, $7 a sample, full cost recovery for times 1,000 times for every single day for each person to get the actual number of the actual quantification of every single organism. And we don't have good primers, good probes to actually detect each organism. That's the only way we would have been able to do quantification before. We developed a new approach with Carsten Zengler, Rob Knight, myself here at UCSD that facilitates this at scale using existing technologies to get absolute quantification of every single organism. What does that look like? Well, it looks like you have say 500,000 cells per mil of fecal material of Bifidobacterium longiminfantis in your stool, or your Faecalibacterium pranisii abundance is close to 1 million cells per gram. That's great, and that's changing day to day. You've got 1 million on one day, you've got 1.5 million the next day, you've got 500,000 the day after. What does that mean? Well, now I've got absolute abundance of cells. I can now tie that absolute abundance, irrespective of anything else in the data, to features like your immune health, your blood chemistry, your blood pressure, as Kevin was talking about earlier, your brain waves, anything else we have, I can tie it to because it's now absolutely quantitative, like the rest of the data in your clinical database. Yeah, so this is a game changer for that particular area.
Awesome. We have a few more questions that came in. One that asks, are tests from Biome and the likes useful today, given where we are with our current data and biomarker knowledge?
Oh, do you want me to answer that honestly? Do you want me to take that, Kevin?
Yeah, answer it. Yeah, but
Biome is a company that's leveraging what they tout as a innovative approach, which is essentially using proportional RNA-seq, where they just sequence the metatranscriptomic data out of stool, which they tout as being a mechanism to identify exactly what the organisms are actually doing, not have the potential to do, but are actually doing in a sample. The problem with that is it's not exactly true, right? The RNA degrades quite rapidly after it's been produced, and the transcriptional response that they're seeing in the organisms are the transcriptional response of organisms being compressed into a stool pellet inside the colon as they go from a relatively anaerobic to a more aerobic system. So it's actually not very indicative of what's going on inside the gut. It's more indicative of organisms being stressed as they're coming out. And we've demonstrated that, and hundreds of other colleagues have, and have tried to tell them that their data is not particularly responsive to what they're saying it is. But these are some of the approaches which are maybe being mishandled by some of these companies. I'll maybe stop there.
Yeah, that's fine.
A few more questions, and then I think we will close it out. Alan asks, how does one communicate this complexity to consumers? Any suggestions? It's a bit broad, so Alan, I don't know if you have any other context you want to share, but we could answer it more generally, either Jack or Kevin, and then you could follow up if that's kind of not what you were looking for.
I'd love to answer that because it's really simple, right? You don't give them the complexity. When you look at heart rate, you look at BPM up on the screen. It's a number, right? The actual systolic and asystolic fluctuations in the heart rate calculations that go into producing that number are incredibly complex, right? The heart rate itself, not just listening to it with a stethoscope, involves a huge number of variables, and the human ear can't really pick up on, not very easily, and computers are now starting to disentangle. But the complexity in just your heart rate is vast. So we alter what we're actually able to say. So for example, in a new company that one of my colleagues has just launched, they're able to take the blood microbiome and use it to predict cancer stage, right? But they don't actually communicate that to the doctors even. They just literally stipulate a area under the resource curve variable, which says that that microbiome is predictive of this. The machinery behind the situation is not necessary. We need to tell people what they need to do, what this number actually means, not necessarily what it entails. Because I don't understand how my electric car works, right? Not properly. So I don't need to. I just need to know it drives.
Yeah, totally. That's a great analogy.
Exactly.
Well, I know we're a few minutes over time, and really want to thank everyone for joining us today. But especially want to thank Jack and Kevin for being so generous with their time this morning, and you all for answering asking such thoughtful questions. We will continue with our Future of Food series. This was our third one. And so please stay tuned to the Blue Stein socials for an announcement for our next one. And thank you both, Jack and Kevin.